Hi, I am Hasif Shah and today we are going to talk about DNA sequencing. Prior to mid-1970s, no method existed by which DNA could be directly sequenced. DNA sequence includes several methods and technologies that are used for determining the order of the nucleotides. Adenine, guanine, cytosine, and thymine are the key players in DNA making of the DNA strands, and these are the codes. By combining three nucleotides, you can make a genetic code. So adenine, guanine, cytosine. If you read it in that frame, it is called a genetic code, and it encode for a specific amino acid. So why DNA sequences DNA sequences was important, or their sequencing of DNA was significant? Because knowledge of DNA sequence, genes, and genome has become indispensable for basic biological research, such as diagnostic. If you want to diagnose some diseases which are related to uh, genetic disorders, you must know the sequence, and if you know the sequence, you will understand that from where the diseases origin, by which cause disease is originated, because there must be some mutation in a specific DNA sequence or gene sequence which is causing problem or genetic disorder. In biotechnology, if we need insulin, so we can insert it in bacteria which will produce insulin. So for insulin producing, we need insulin gene. So for gene, we must know the sequence. So for that purpose, we must have the knowledge of the sequence, and it comes in the under the umbrella of biotechnology and forensic biology. As you know that in forensic biology, what we do, we we try to capture culprits. So for that purpose, we collect samples of the culprit at the spot of crime, and we match it with the DNA. So that also need the sequencing of DNA. So it is very important. DNA fingerprinting, as you will be familiar with, comes in forensic biology. Okay, biological systematics. So biological systematics also needs DNA sequencing. The advent of DNA sequencing has significantly accelerated, improved, or you can say that fastened, or you can say that abrupt increase in biological research and discovery. So DNA sequencing, there is a bit of history. We will touch it. The first DNA sequencing were obtained in 1970s by academic researchers using some very laborious methods based on two-dimensional 2D chromatography. Following the development of dye-based sequencing method with automated analysis, DNA sequence has become easier and order of magnitude faster. So they were trying for the best. Here comes these three methods. One is on another slide, which gain very popularity, and today's work is also based on these three. So Maxim and Gilbert method. We will just talk about Maxim and Gilbert method today. Today's video. Next video I will make sometimes later. So in Maxim and Gilbert, we have some chemicals which cleave specific nucleotides. So some chemicals just cleave adenine, some chemicals just cleave cytosine, and other chemicals cleave just. So every adenine, or some chemicals cleave both adenine and guanine, partially or completely. So on the cleaving of the adenine or guanine or cytosine or thymine nucleotide, which are labeled with radioactive phosphate. So we are going to talk about it in detail in next slides, but I may give you a very simple overview that we add adenine, and if adenine is which is labeled with uh, radioactive phosphate. So if adenine is added and chemical cleave it, chemical cleave that strand there, and gives, we will get some radioactivity. So we will understand that there is adenine added, and in a template strand there is adenine, so we can sequence in that way. So we will talk about in next slide. Allen, Maxam and Water-Gilbert developed a DNA sequencing method based on chemical modification of DNA and subsequent cleavage at specific bases. The method required radioactive labeling at 1,5' end of the DNA, typically by a kinase reaction using gamma 32 ATP, phosphate ATP. Purification of the DNA fragment to be sequenced. Chemical treatment generates breaks at a small proportion of 1 or 2 of the 4 nucleotide bases in each of 4 reactions. So let's see. Chemical targets in the Maxam-Gilbert DNA sequencing strategy. Dimethylsulphate or hydrazine will attack the purine or pyrimidine. Okay, in respective way. Dimethylsulphate will attack purine and hydrazine will attack pyrimidine and cleave it respectively, and piperidine will cleave the phosphate bond at the 3' carbon. So dimethyl sulfate and piperidine alone will selectively cleave guanine nucleotides, and dimethyl sulfate and piperidine in formic acid will cleave both guanine and adenine nucleotides. Remember that, it will help you to understand. You must remember these 4 points. Hydrazine and piperidine will cleave both thiamine and cytosine nucleotides. Hydrazine and piperidine in 1.5 mole NaCl will only cleave cytosine nucleotides. You can see it in the diagrammatic form too. So now sequence. The Gilbert manual sequencing scheme, the target DNA is radiolabeled and then split into the 4 chemical cleavage reactions. Each reaction is loaded onto a polyacrylamide gel and run. Finally, the gel is autoradiographed and base calling proceed from bottom to top. If C is added, so there will be a stoppage of reaction, because chemicals will cleave it here. So we will get a radioactivity on the film. So we will know that there is cytosine added. So cytosine make hydrogen bonds with guanine. So we may say that on template there is guanine. So it is enough for today. We will talk about Sanger method in next video. So I hope you understand. If there is any help needed, I am here for you, and will help you in your studies and your understandings. Thank you.